Most thymocytes express receptors that cannot interact with self-MHC, and these cells die in the thymus. Bone marrow chimeras and thymic grafting provided the first evidence for the central importance of the thymus in positive selection. But more detailed investigation of the process has used mice transgenic for rearranged T-cell receptor genes. The rearranged alpha and beta chain genes are molecularly cloned from a T-cell line or clone whose origin, antigen specificity, and MHC restriction are well characterized. When such genes are introduced into the mouse genome, rearrangement of the endogenous genes is inhibited, and most of the developing T cells express the receptor encoded by the transgenes. By introducing T cell receptor transgenes into mice of known MHC genotype, the effect of known MHC molecules on the maturation of the thymocytes with known recognition properties can be studied. It is routinely found that T cells bearing a transgenic receptor develop to maturity only within a thymus that expresses the same MHC molecule as that on which the original T cell receptor was selected. In this situation, the developing transgenic T cells can be positively selected. Such experiments have also established the fate of T cells that fail positive selection. Rearranged receptor genes from a mature T cell specific for a peptide presented by a particular MHC molecule were introduced into a recipient mouse lacking the MHC molecule, and the fate of the thymocytes was investigated by staining with antibodies specific for the transgenic receptor. Antibodies to other molecules, such as CD4 and CD8, were used at the same time to mark the stages of T cell development. It was found that cells that fail to recognize the MHC molecules present on the thymic epithelium never progress further than the double positive stage and die in the thymus within 3 or 4 days of their last division. And that's most thymocytes express receptors that cannot interact with self-MHC, and these cells die in the thymus. Have a great day.